Kwang-Hyun Cho, a systems biologist and bioengineer from Korea, has piqued my interest because of his advancements in cancer research. Cho started his path in the world of electrical engineering, earning his doctorate in electrical engineering from the Korean Advanced Institute of Science and Technology, where he is actually a professor in the Department of Bio and Brain Engineering. He also directs a lab for systems biology and bio-inspired engineering. While he does all that, he is also an entrepreneur in the sense that he is a founder of the company BioRevert Incorporated, which works specifically with cell reversion therapy in order to revert cancerous or diseased cells back into normal, healthy cells. And Cho is a normal human being like the rest of us. He has his own interests, and particularly, he likes to paint. Check out his work, he does a good job. Hyun Cho and his team have become pioneers in cancer research and development, coming up with new key ideas like cell state reversion, as we mentioned earlier, about bringing cells who have transformed into cancerous or disease-ridden back into their original healthy states. Control theory in biology, which takes concepts from engineering and applies it to these biological systems. Things like feedback loops, stability analysis, and network controllability. Critical transition theory refers to tipping points when cells are irreversibly transformed into diseased states. And then the therapeutic innovation ties right to cell state reversion. And so instead of killing or destroying the cells that are cancerous, we're actually reverting them back into healthy ones. The article, Discovery of a Kernel for Controlling Biomolecular Regulatory Networks, written by Cho and his team, dives into the idea that a certain section of nodes can actually determine the entire regulatory network of a cell. The idea of the control kernel is that it is a select group of nodes within a regulatory network that will influence a chain reaction amongst the rest of the network, where we can influence that one control kernel and influence the entirety of that network. A crash course version of how they defined the control kernel as being real or not started with a construction of a network that resembled a model organism's biological regulatory system and the interactions it has. Then moving on to define what a normal or healthy cell state would look like. They would then assign values being 1 or 0 to represent on or off and simulate the system's evolution. Then by using a genetic algorithm, the model searched for the smallest combination of pin nodes that guaranteed convergence to the target attractor, aka the healthy or correct cell type. Then the system is repeated over and over across multiple known biological regulatory networks to see if it is matching. Figure 1 gives us a great visual representation of these control kernels being found and utilized to impact the entirety of their regulatory network. This means that cellular behavior can be influenced or reprogrammed by targeting only a few key regulators, and it really does open the door for therapeutics like revision therapy. With the new wave of technology with AI, the next steps are going to be integration with AI to help with these system analyses. In vivo validation, using animal models and essentially mapping their regulatory networks and seeing how we can influence those control kernels. And right now there isn't much going on clinically wise, so what they are trying to do is advance that so that we can help, or they can help, more people. Thank you all for watching and appreciating Kwang-Hyun Cho's work with his team, and I hope you all have a great day.